Today we'll be presenting on Chapter 5 of Improbable Destinies. My partner is Anishka, and I'm Lisa. So to begin, I'll start with a general overview of the chapter's content. One of the major points presented by the author is that although we can't turn back the clock and observe the process of evolution in the past, scientists are able to conduct evolution experiments. By doing so, they can test for evolutionary determinism, which is a concept that gene frequencies change in a directed process, in contrast to change due to random chance. Several studies were outlined in this chapter, including Haskins' lab predation studies, Endler's dual experiments, Resnick's life history study, McGurn's behavior experiments, and Osteen's experiment on guppy response to predator interactions. Endler's guppy study, in particular, involved both observational and experimental components. It is mentioned in the chapter that a lot can be gained by observation of natural phenomena, even in the absence of the manipulation of the population. Observational studies provide a source of the hypotheses that can then be tested by experimentation. In this way, observational and experimental sciences go hand in hand. Through both observation and experiments, it was concluded that strong natural selection and selective pressures will lead to rapid evolution. In the case of the guppies, it was shown multiple times that their evolution was predictable given selective conditions. One last point made in the chapter is in regards to the ethics of experimenting in nature. There can be harmful effects that stem from moving populations of fish around, in that this can disturb natural ecosystems and the way they function. However, arguments can be made both ways in favor of scientific progress or preserving nature as it is. So here is a quote from the author regarding the ethics of altering nature for scientific gain. At its heart, this is a philosophical difference about scientific progress and the sanctity of nature in a changing world. There is no objective right or wrong, just a difference in opinion. Although these experiments can be intrusive at times, these guppy experiments often mimic the natural movement of guppies and contribute to the expansion of scientific knowledge.
In 1946, Carol Haskins traveled to Trinidad to observe guppy populations in streams with waterfalls. And while he was there, he noted that male guppies which lived in low predation areas upstream of the waterfall were very colorful, while guppies which lived in the higher predator areas which were underneath the waterfall were dull in color. And when Haskins collected samples from both waters and placed them in his own lab aquarium with Trinidadian predators, he noticed that the colorful guppies died out at higher rates. And these observations showed a clear correlation between the predator presence and coloration of male guppies, as the presence of predators decreased the coloration of the male guppies. And later on, John Endler used Haskins' observations to formulate a hypothesis that over time, the guppy populations, which are exposed to higher levels of predators, will get increasingly duller, while those exposed to little to no predators would become more colorful due to sexual selections, aka the females will select the more colorful guppies when there are no predators. And what Endler found by carrying out his double experiment was that when colorful spotted guppies from low predation areas were placed in high predator experimental conditions, he observed an eventual reduction in average spot number and color after a few months. Likewise, when the dull guppies from high predation areas were placed in experimental conditions with no predators, he observed a gradual increase in spot number within just a few months. And to go into a bit more detail about this dual experiment, he basically took guppies from the streams and placed them in a lab setup, which he made himself, as well as a stream in Trinidad, which had the predators, but it did not have the guppies. So he basically introduced guppies to that stream. And why were Endler's experiments so important? Well, there's three main reasons. One, they showed that guppy evolution was predictable. He could subject the guppies to any sort of condition and kind of determine what the results of that would be. It showed that when natural selection is strong, evolution occurs rapidly, which in the case of these, it occurred in a few months. And the experimental method can be used to study evolution both in the lab and nature. So what's a medical application? Prevalence of certain diseases in areas of the world can lead to selection for phenotypes which confer resistance to that disease. So for example, people with sickle cell anemia have some degree of resistance to malaria, And when you compare maps showing the distribution of the HBS or sickle cell allele and the prevalence of malaria around the world, there's a lot of overlap in certain parts. So what does this show? It shows balancing selection for heterozygosity, meaning that the prevalence of that allele would be higher in certain areas than if it were just due to like random chance. And in high malaria areas, people who possess one sickle cell allele and one normal hemoglobin allele will have increased fitness compared to those homozygous for either allele. So that's an example of heterozygote advantage because heterozygotes will have normal blood cells, but they'll also have the resistant sickle shaped cells. And keep in mind that having both alleles for sickle shaped cells will mean you won't have any normal cells, and that would be disadvantageous.
One medical application of this chapter is that arbitrary boundaries in place can lead to differential outcomes for various groups. As was evident in the multiple guppy experiments, many character traits varied depending on if guppies lived upstream or downstream, or in the presence or absence of predators. In a similar fashion, where an individual lives and the barriers said individual faces can lead to very different consequences, such as inequitable access to healthcare and resultant disparities between communities. In one article published in the Harvard Gazette, the author Colleen Walsh explains that certain communities, particularly the African American community, face increased risk of contracting the novel coronavirus COVID-19. Many of the health disparities are the result of long-standing racial structural inequalities, and residential segregation is one of the primary drivers. Another article, Wang 2010, explains that locations and quality of healthcare providers vary from place to place. The graphic presented on this slide shows the relative risk of a late-stage breast cancer diagnosis across zip codes in Illinois, with darker regions corresponding to higher risk. The inlet graphic shows that southern Chicago has the highest values of relative risk. This corresponds to a high concentration of vulnerable people living in economically disadvantaged residential areas. So, where one lives can be a determining factor in the quality and level of access to healthcare one receives, which is a harsh reality that many face today.
So why are there different outcomes for different groups? Well, looking at the past few scenarios, in the case of the guppies, the presence or absence of predators affected their phenotype. In the case of sickle cell, the allele is more prevalent in areas with higher incidences of malaria. And with regard to the communities in Illinois, the areas in which one lives in affects their level of healthcare access. So the main thing in common with all three of these situations is that members of the same species, whether it's humans or guppies, experience different outcomes because they're subjected to different conditions. So these different conditions will basically influence how these organisms will live and develop.
One important question to ask is, how can structures and processes be changed to support more equitable outcomes? In terms of experimental ethics, we can support more equitable outcomes by taking a variety of approaches. First and foremost, we need to take into account the intrinsic value of other species, not only their value in contributing to scientific research. Organisms have inherent value that is not derived from the services they provide for humans, so we need to shift current human-centric views. With this being said, studies should be conducted to investigate how interventions of natural processes, such as introductions of species to regions they are not native to, affect organisms directly and indirectly. Another way to support equitable outcomes is to pass legislation that limits experimental introductions to those that are deemed necessary for scientific advancement or not harmful to the organisms. In regards to health inequities that arise due in part to residential differences, there are several solutions that could aid in closing the gaps and making healthcare more equitable for those in disadvantaged communities. One possible solution is to provide subsidies for healthier food choices to expand access to them and decrease reliance on fast food, which tends to be more affordable. Another way to decrease health inequities is to ensure that there are quality and affordable healthcare providers in close proximity to communities who suffer from greater rates of disease or health issues. We can also strive to increase the number of health screenings for certain diseases and cancers to decrease the relative risk of a late diagnosis. Changing these structures and processes will contribute to making medical care and, as a result, health outcomes more equitable for all.
So for the discussion questions, we have these 10 questions. And for question 8, I'm going to clarify by saying that by selection for a certain allele or trait which is advantageous against a certain pathogen, I'm thinking about diseases which would be similar to sickle cell. So what kind of allele would be selected for to give a degree of resistance to some sort of condition? These are the works we cited. Thank you.